sounds like you might have, I mean, I had endometriosis, you might have this as well, and that could be causing some of your problems. And sure enough, she told this to her doctor. Her doctor was like, wow, we didn't think about that. So they actually went in kind of the same way and poked around, and guess what? It was. She had so much endometrial growth around her left ovary that, so this side, her left ovary, that what it was is it had ruptured her ovary. Okay, and that was the severe necrosis, like the decaying matter, and that was the pain and the infection and the high fevers and the passing out and stuff like that. So they removed the ovary, they roto-rooted her, and then she's fine now. So females, if you have very severe abdominal cramps, okay, in kind of abnormal period sort of deal, and you might think about that. Okay, and it can influence your fertility. Yes.
Does the endometrium regrow though once they took it all out of you?
Yeah. So the doctor told me afterwards. He said, "Well, you might want to, after you're done with this whole pregnancy stuff, you might want to go on the birth control pill, because that helps like lower your hormones and lowers the endometrial lining. Some of them prevent endometrial lining from building up and that sort of stuff, which can prevent pregnancy. So they had actually advised that I go on the birth control pill after we were done being pregnant, and then all that sort of stuff." Which I didn't, only because of the birth control. I have migraines, and so it causes migraines. So I know right now that my insides are getting corroded again. Jumping up again. Okay. All right. I'll take 2 more questions, then we gotta move on. Yes, Aaron.
So do you occasionally have to go get it cleaned out again?
Yes. If that will, in the future, if I notice like the kind of the symptoms that my sister was having, or if I'm having that sort of age, I will probably have to be cleaned out again.
Okay, so I have 2 questions. I'll just ask one.
Okay.
So if your fallopian tubes couldn't like suck up the eggs, were there like a buildup of dead eggs?
Well, they would be the body would find them and they would be engulfed. Yeah, they would just be eaten by the immune system. This is a dead egg. Let's take it. Yeah. The other one, real quick.
So if the endometrium was growing on the outside, was it also growing on the inside?
Yes, I still had periods, but they were irregular. Like 1 month it'd be really heavy, and the next month it'd be really light. That might have been the month where it was growing on the outside instead of the inside.
I also switches between. Okay.
Yeah. Who knows? They don't know why endometriosis occurs. So it was just, so if you have that irregular pattern, not you, but if you have a period, Alright, so morphogens, this is another name, okay? It is kind of referring again to any substance that provides position information. So these are, what's the deal with this is, what do you notice in the word morphogen? More. Okay, so this is really in relationship to morphogenesis. So these substances are usually located along an axis, okay, or will establish the axis. There's a gradient. Remember what we talked about with gradients and transport, right?
Yeah.
You have passive transport and you have substances diffused down their electrochemical gradient, so there's a high concentration below. So the cytoplasmic tumors, remember the picture I showed you where I said look at where the red dots are? Mm-hmm. There's more red dots in one area versus another. So that would establish a gradient. Where there's a lot of those red dots, you would have a lot of production of proteins. So you'd have a high concentration of proteins in one area for a particular, transcription factor, and not a lot in the other. So that created a gradient, all right? And it will set up the axes. And you'll see that in a couple of pictures I'm going to show you in the next few slides. And again, most of these morphogens are proteins, which are created by the egg polarity genes, okay? They're going to cue positional information. They're your cytoplasmic determinants that will turn on certain genes and make those proteins. Like I said, most are transcription factors. So this is an example that they will go through in your book if you wanted to read about this. I'll just give you the short version because of time. Basically, if you look here, you're looking at the gradient. This is a Drosophila embryo. And they have this bicoid gene, which they've done a lot of different experiments on. And they know that it makes the bicoid protein. And they've tagged it with those fluorescent markers. Okay, and they were able to see the production. And And they know that in one region you'll have a lot of this particular protein, and it kind of degrades or goes down as you go across. And that helps establish the different ends. This is the anterior end, this is the posterior end, for you. Okay. And then that's also this picture here. Okay, you can see. Do you see the different segments? All those different kind of white patches, those are all, it's saying that with each of those, remember when you were doing your coloring, you would have the different like lines, okay? That's the same, there's different segments there too, and they are also establishing, oh, this is the anterior portion, this is the posterior portion of the segment. Overall, this is going to be the head, and then each section is going to have a front and a back sort of deal. Okay, so I don't have to spend much time here, I think, because you did the article. I just want to make sure you understand homeotic genes. Alright, these are what we would collectively say are the master control genes, right? Your article talks about an architect. What does that mean? Design a building of some sort, a house, a major building, a skyscraper, whatever you want to call it. They lay out the plans, right? They say, this is what we're going to build, this is how big it's going to be, we're going to build this portion first, and they make what are known as blueprints, don't they?
Mm-hmm.
Right? Okay, and those blueprints have to be approved, several different levels and layers and all this sort of stuff. And all of it has to come together in a coordinated fashion in order for somebody to come along and actually start doing the building. And that's kind of what these genes do, is they lay out that master plan. These are the homeotic genes. Put a leg here, put an antenna there, right? They control pattern formation in organisms. And any mutations in these genes, you get the wrong parts in the wrong area. And did they have the picture? In that, I know it was kind of black and white. I have the actual picture I can show you. It's really cool. Where that one Drosophila and you,
140.
Oh, okay, we might have. So I might show you a video where you can see as well the Drosophila that has the legs where the antenna should be. You're like, oops, you put that in the wrong part. Okay, that wrong part in the wrong place, sort of deal. So this is the picture. So these are supposed to be the antenna. You can see they're compact. They're there for like feeling, kind of sensory, but oops, we got legs. Okay, this one is really cool. This is Drosophila as well. And this, what's wrong with this or that mutant?
Two sets of wings.
Does that make them fly faster?
Oh.
Got an extra, it would be like us getting an extra thorax. We'd have a heart and a lung here, and then a heart and a lung here, and then we have a abdomen. And we have arms and arms. We have 4 arms. 2 arms, 2 lungs. It sounds kind of cool, but guess what? Finding a shirt will be really difficult. You have to learn how to sew. This actually does not help the fruit fly any better. Okay, they don't quite know how to coordinate those 2 sets, so they don't fly as well as you would think. Oh, they're super flyers, right? No, they don't fly as well as their normal, non-mutant, Drosophila brothers and sisters. Okay. So this, you can see that extra segment. Oops, God. So something, obviously the master control gene that said develop deception got 2. Okay, those about. Now, what you read about yesterday again in the article talks about Hox genes. Hox genes stand for homeobox. Okay, and it's a region. Okay, the homeobox is a region on these homeotic genes. I tried to draw the picture right here for you. Okay, and I'm not the best artist, but basically what happens is this particular region, which is the Hox genes, which is short for homeobox and Hox, right? What it codes for is as a region that homeodomain, okay, which then says, okay, that's where the protein, like a transcription factor, is going to bond to the DNA. And remember, we know that is incredibly important for transcription. If you don't get transcription, you can't express that gene. So the Hox region is really important. And so what we have noticed is that that particular region is highly conserved between species. What does that mean? Highly conserved between species.
that mean that it's within all the organisms?
Yes, that same portion, like this little coloring activity that you did. This is why we did this, right? If you look, that she's got purple for A, whatever color you have. Notice that A is found in pretty much all of them. Yeah, it's not in home oxy, but you have two other copies in the others. It's there, and each one of those is coding for a very similar region, right? Okay, in the development of that organism. And they have done multiple studies where they have found these homeotic genes. They have extracted this particular area. They've sequenced it. They've sequenced the entire genome. Okay, and they've looked at this particular region, and it's found that it's almost identical in every species. It's so identical that I can take a fruit fly Hox gene and put it in a different species, and that different species will be able to form like that species, but using the Hox gene from the Drosophila. Okay, because it codes for, you're going to be this position, we need to turn on these now, and it allows for that particular gene to be transcribed. Right, so it's highly conserved. And you can see that in this picture here too. Again, the similar colors being found in the fruit fly versus the mouse, like you guys did with the coloring. And you can see similar areas of development within those organisms as well. Okay? And it's very much conserved because it's so critical to the formation. We've got to get that pattern down. If we don't get the pattern down, the organism can't form properly. Okay? And this is just the same kind of picture there for you. Alright, so I'll quickly show you that video. It's only 3 minutes, so we'll be good. And then we'll be done for today.